Hello, hello. Happy Wednesday, everyone. Welcome back. It's Thursday till an hour ago. Well, good news. Thursday's tomorrow. So you're day back. Cricket, what's up? Courtney, Clandestine, Liz, Docky, Donkus, Mop, Villa, hello. Okay, so today is Wednesday. And so that means we are back on our pre-chat chats grind. Um Going through our synoptic collection of beetle families. But today, we're actually making some good forward progress. You see, for the last 12, 16, 17, 18, 19, 20. The last 20 families, which is probably like 5 or 6 weeks of pre-chat chats, have all been within the superfamily Tenebrionoidea. Last week, when we wrapped up with the false longhorn beetles, that was our last Tenebrionoid family. So we're moving on to a completely different group now. We're going to learn first, to the best of our ability, because it's not a super well defined group. But we're going to learn a new set of characters that all of these families should more or less share. And yeah. So today we are going to start working in Cleroidea, Cleroidea. And even though this is not the first family in the list, it is the one that this group is named after. So I think it's probably appropriate to start. We're going to be starting with the family Clarity. Like that Zed song. Um, you are now here. Granola, hello, hello. There we go. All right, so we'll zoom out just a bit. So this is a checkered beetle, the family Clarity. So this is the largest group in the superfamily that we're going to be talking about. And so it makes sense for us to start with this one because there's a lot of diversity here. Some things you've maybe seen before. And also typically like the the standard more or less for the whole superfamily. So these are called checkered beetles because a lot of them have a bright contrasting color pattern, but not all of them. This one is pretty checkered, so we're okay there. Then we have this guy, which is brighter in color, but still checkered. Then we move over here, and we start to lose the plot a little bit. This one, not very checkered. Moving down here, we've got some contrasting colors with that bright orange pronotum and black elytra, but no checkering. And then lastly, the till lines here. This is not very checkered. That's kind of a brief overview of the diversity in this group. This one here, Enoclaris. This is this is like the default Clarid when I think about this group. There are a lot of these that have this colorful band or little colorful spots on the elytra like this. So this is like default Clarid in my mind. Something else that you might have noticed that is shared amongst most of the things in this group is that this thing is quite hairy. So all up here on the head and the pronotum, all the way down here through the elytra and the legs, and then basically down all the way down the back. So most of the things in this superfamily do have some sort of fuzziness to them. Now, not all of them. Remember, these things are rules, but they're more of guidelines. Because evolution does some really weird stuff, and it's hard to put insects into meaningful boxes because there are always little bits that are exceptions to what we would expect. Whisker pickin'. Interesting. So for this group, Claroidia is not the biggest of the beetle groups. There are only about, I say only, but there are about 10,000 species of this group worldwide. There was a big treatment of them just a few years ago. So we actually have a pretty good handle on who's related to who in this group. And one of the other characters that we have looked at historically for the tenebrianoids was the tarsal formula. Now, this might come back to bite me, but most of the families in this group have 555 tarsal formula. So, in theory, the front, middle, and hind legs should all have the same number of tarsal segments. Now, don't get scared because the tarsi are lobed. They do have sort of the wide toe pads that in other groups occasionally conceal additional secret segments, but not here. Rotilla ain't counting those. Okay. So here, this is a middle leg that we are at the moment. There are 1, 2, 3, 4 that are lobed, and then the long fifth that has the claws attached. Easier to see down here on this hind foot now, where there is 5, 4 is lobed, 3 is lobed, 2 is lobed, 1 is lobed. But you can see that even the underside of this group is also quite hairy. And so again, that is a not completely consistent, but decent thing to potentially look at for this group. So everything in this group, even the more plain looking ones that could be confused with a tenebrionoid, if we count the tarsal segments, we know it's not a tenebrionoid right away. The only I won't say that. But some other good characters now for this family specifically is they've got some pretty bulgy eyes. Dare I say, bug-eyed. Um, but they have relatively large eyes that kind of do stick out to the side of the head. They are also pinchy, correct. As larvae, these are going to be in wood, but the larvae are not there feeding on the wood or feeding on the fungus like some of the other groups. Clarid larvae are predatory on other insect larvae that are in the wood. The adults are predatory as well. Um, mm, dare I say most of the time? I would say some of the time. Because there are other groups, like that really bright metallic one that we looked at, that given its coloration and its pattern, you might say, "Oh, that's bright and colorful. That might live on a flower," and in fact, it does. So some clarids are predatory, and you'll find them running up and down plants, and tree bark and things like that, feeding on the other insects. While others, some of the brighter, more flashy colored ones, are going to be on flowers, eating pollen and things like that. So, relatively big eyes, checker patterned most of the time, elytra, hairy, big chompers, not always this obvious because we're also looking at an underside angle right now, where we have a very clear view of the head. Um, but then they also have these clubbed antennae. Roxers, thank you for the 6 months. Uh, oh, you've got your founder badge. Shoot. You're the first one to unlock the new sub badge. Who was 6 months? What did we give for six? I think 6 months is the earwig. Let me double check that. 3. Yeah, 6 months is the earwig. So you have the new earwig badge, but you also have your founder's badge. So I don't know how to do anything about that. But thank you for the 6 months. It's crazy that it has been 6 months already. So there's my obvious. Yeah, I don't think you can. I don't think you can hide founder, which is a bit of a bummer. But I appreciate the support regardless. I think Roach is pretty close behind you. So if she shows up tonight, we'll maybe get to see it. Um, but anyway, back to our clarids. Clubbed antennae, sometimes, not always. This one is pretty obvious. Remember last time when we were talking about what an antennal club is? It doesn't always have to look um it doesn't always have to be like a big ball on the end of a tweezer or a tweezer. What did I say? Stick. I meant to say twig. Um, so like butterfly antennae are also clubbed, and their clubs more or less look like this: long, straight antenna with just a knob at the top. These are also more commonly called capitate antenna because there's just the little thing up at the cap. While we're here in the full screen, let's just give one more minute of appreciation to our well organized pin box that we did on Sunday. 4 hours of pin organization. The big one is put up over there. But this was Tortilla, you missed it. Um, this is the little box, but we also have the big box. So we didn't end up counting all of them. That would have been a horrendous activity for everybody involved. But it took about 4 hours, and we organized all of our pins, and we weighed it, and it is more than half a pound. It is more than half a pound of insect pins, which I think is a ridiculous number to have. But we do. So that was Sunday's stream. I had to do it anyway, so I figured why not go live. Thank you for streaking, Dan. 25. God damn. Thank you. Thank you for the support. 25 feels crazy. So going back to here, I saw Jules say that it was easier this week than last week, which I totally agree. Last week we were talking about antennal clubs and how they're not always like super duper obvious. The thing is, you need to see where the antennal segments change shape, and sometimes it can be quite subtle. But as Jules said, these ones are much, much more obvious. Gabby, thank you for the follow. And Nicole, thank you for the lurk. Thanks for stopping by. These ones are a lot more obvious, right? You can see the super clear transition where it goes from normal antennal segment to kind of broad triangle. Then you have another broad triangle and you get this nice teardrop shape. That end antennal segment doesn't look like that in every single group. But we'll take a look at some of the other modifications here in just a bit. But clarids in general have pretty distinctly clubbed antennae, and it is a lot more obvious than the ones we were looking at last week. Like for instance, the bright metallic one that lives on flowers that we caught a glimpse of earlier. Look at that antenna. Now that's a club if I've ever seen one. It's an oar. Yeah, so this is, you want to fight. Yeah, he does kind of look like a put him up, put him up. So there they are on that one. Echinoceron doesn't really have good clubs out. This one's got some fun clubs. This is what I think it is. Kyriadella. This is a bad club. It's a cool antenna, but it's a bad club. Uh, because this one is like maybe only one segment and it's just the very end. What I thought this was, or what I was looking for. So this must be a female Curiessa, because this is the antenna that I associate with this genus. Um, and again, this is a club. That is a crazy club going on. But this is a male, and the one I have must be female. Again, male individuals will oftentimes have more exaggerated antennae. In moths, they're a lot bigger and fluffier. In longhorn beetles, they're longer. In scarabs, they have those really big, like antler looking things. And all of that is to add additional real estate for chemosensory receptors, which will allow them to pick up on the chemicals and the pheromones given off by females. So if you ever see an insect with exaggerated antennae, it's I won't say always, because again, insects do weird stuff, but like 99% of the time it's going to be a male, and all the time why it's that big is because they're using it to sense out females. So the one we have, going back, our friend here, this is that same genus, but again, this is a female, and she does not have that super, super big club here at the end. Now, that's all of the ones in our display box here, kind of our representatives, except for the Tilline. Now, this one is, oh, this is a flail mace. I would love to do like an insect based armor set or weapon set. That would be really neat. So this is also a Clarid. And that kind of feels bad. It has the narrowing pronotum. It has the relatively bulgy eyes. It's hairy. It has the 444, or nope. It has the 555 tarsi. But if we come out here, there is no club of which to speak. And that is typical of things in this subfamily. Up until, I actually don't know when it changed. I don't know when they got included, or if they specifically were when they got moved. But this subfamily used to be its own family. And so it keys out in its own grouping, based on its essential lack of characters that would put it in anywhere else. Does it have a function to have bigger clubs? Yes. So the males will often be the ones that have those larger clubs to have more surface area to pick up pheromones from females. But if you mean like between families or between things in a family, like why this one doesn't, while the other members tend to. Um, it's usually down to like a way of life adaptation in some way. Um, so for instance, male fireflies, which we haven't gotten to fireflies yet, but male fireflies will have really, really, really big eyes. And if you think about it, that makes sense because what do we know about fireflies? They use their lights to flash and communicate to try to find other members of their same species. And so, if you're a little beetle and you're looking for a little female beetle, you need to be able to see that visual flashing signal to figure out where, in this giant sea of other fireflies, you need to be able to see and discern your species' flash pattern. And so, because of that, males will have these really, really big eyes to be able to make sure that they can see the females. But in fireflies that don't flash, and there are plenty of those where they do not glow as adults, um, the males have smaller eyes, but they have larger antennae. And again, that's just because those non flashing fireflies use a more standard chemical signal than a light signal to attract mates. I do not know the difference between like how this subfamily of clarids versus the others find or attract mates. Um, to say like that it's definitely the difference, but that is one possible explanation. Lame ass fireflies. What do you mean? All fireflies are good fireflies. But I do say that as the bias of someone who grew up with and worked with fireflies for a while. And I certainly did miss them out west. But this is a perfectly valid clarid. It checks all of the physical character. Well, it checks a lot of the underlying uh traits that we think of with clarids. But some of the definitions obviously just come down to can also come down to like some genetic information, which this does. Again, these all do what am I saying? All of these do uh line up genetically. They all are related to each other, and we're pretty confident about that. But we got to keep our leaf litter. Yes, yes, you do. But let's grab some of these out, and we will talk more specifically about clarids and maybe some of the stuff that they're doing. I'm going to save the bright, shiny guy for last because I know you guys are going to want to see that one. So first we have the tilline. This is Cimatidera spa and doesn't really give normal. I think it's a normal beetle. Typical clarid vibes. Then you've got Enoclaris, which again is what I think of as like the default clarid. This body shape, especially clubbed antennae, big eyes, and then I don't think I talked about this, but the pronotum, so that neck piece, is narrowed towards the back. So you can see on this one that where the pronotum attaches to the head is wider than where the pronotum attaches to the elytra. And that's another thing that you can see here, even if it is a lot more subtle. The pronotum is narrower at the base than it is where the head attaches. And that's another thing that not all the time, but is another clue into the IDs with this group. Then we saw this one. This is that female Cariessa. Oh, pickle! Thank you. Let me get you get some fancy toes. We'll skip over here, and these are going to be of the big shiny guy. Back it. Oh, hell yeah, Vic. Just in time. Look at that. Daily dose of toes. Again, this is a hind foot. So this is a pretty clear 5 segment. We're not worrying about... Oh, toes is an emote. Oh, it's a pink toe tarantula foot. That's awesome. I love that. Thank you, Pickle. Thanks for stopping by. And little teaser. Vic, if you haven't seen this yet. Ooh. Okay. We'll come back. We're going to come back to that one. Ad break? More like sad break. Hire a sort of cool beetle toes and I'll be beetle toes immediately. Okay. I can work with that. I don't have any problem with it being tarantula toes. Pink toe tarantula feet are very, very cute. I just didn't realize it was an emote because I'm watching chat on OBS. But over on Twitch client, it's I get all my... what was I saying? Oh yeah, Curiosal. Okay, so this guy, or gal, I should say, since we checked on that. Again, the pronotum, that orange and black piece there in the middle, basically the neck, is narrowed right where it attaches to the elytra. And this is another bit of an oddball group. This is a Hypnocerine. So it's a separate subfamily than the others. And these have a characteristic shape to them because they have such large eyes. I know having big eyes is one of the things that I said for kind of clarids in general, but Hypnocerines specifically, their head is like really bug-eyed. Short pronotum, relatively small overall, but they also have this elytral gape. So if you look here at the back, get that. Nobody be weird about what it's called. So typically when we're looking at beetle elytra, they come together in the middle and they basically meet all the way down, and then the elytra spread. These ones even if the elytra were closed all the way, you would see that there were two distinct wingtips. So having like this in the key, this would say elytral apices, elytral apices, or the plural of apex, um, individually rounded, is what this would say. So it does give this little swoopy swoopy bit that is characteristic of hypnocerines. And then last, but certainly not least, we have not the beetle. Well, in fact, not the beetle. There's the beetle. Let y'all sit with that for just a second while I try to remember this damn. Trichodes. That's what it is. This this is that genus. And these ones are more often associated with flowers. So you can see that yes, the other clarids were hairy. This one is exceptionally hairy. And part of that has to do with uh pollen. They collect it, they will eat it. They don't store it or anything like that like bees do. But these clarids will feed on pollen. And the larvae of this group are going to be uh, parasites on solitary bee larvae. These checkered beetles will lay their eggs on the flower. The larva then hitchhikes on the adult bee. It will grab onto the tongue or the foot or the leg or some part of the bee and follow it back to its nesting site, where it will then drop off and wait for the larva to develop, and then they will parasitize the larva that it is of the host, which is pretty cool. That's a strategy that we see in blister beetles as well, but these clarids will also do that. Looks like I washed them with your metallic paints. I'd buy another print if you feel so inclined to paint this one. Uh, I'd get another one. But yeah, it is pretty remarkable, and they come in a lot of different colors, honestly. So this species, you have a couple of different color forms that are all the same thing. You can find them on the same flowers. So here's a similar one to ours with the metallic blue and the yellow, but you also get a lot ones that are a lot more red. Have this orange pattern on the back. But you can also see that the pattern differs quite a bit between individuals. Pretty one. So one thing that is in clarity, but is not in my little representative collection for whatever reason, is one of the beetles with the best common name out there. This is a little thing called Necrobia rufipes. And if you look right here, oh, my picture is covering it, so you can't see it. That's pretty funny. This little guy right here is called the red-legged ham beetle. Ham beetle. So this is another atypical behavior-wise clarid because it is not feeding on pollen. It's not feeding on other insects. It is actually a stored product pest. And the types of products that it is a pest of, if the name doesn't give it away, are like stored meats. So salami, ham, any of that stuff. Dried and preserved meat products, you can get red-legged ham beetles on it. In nature, these are going to show up late in the stages of decomposition where most of the other stuff has rotten away and you're basically left with just the dried skin. And so that's how they existed before humans started curing meats. Um, but now we've concentrated all of their favorite foods and oftentimes hang them up in places in bulk together. So this little guy, also kind of metallic bluey green, but it has red legs. So this is the red-legged ham beetle, which is quite fun. I've collected these. I found them on a dog bone in a pet store one time. It was a bone that was wrapped in plastic, and I don't have a dog. I didn't have a dog at the time that this happened either, but I took it up to the front and I was like, "Hey, this is this is gross. You guys have like a weird bug in this bone." And they're like, "Oh my god, I'm so sorry." And I was like, "Well, can I still have it?" And they're like, "You want it even though it's like covered in bugs?" And I said, "Well, yeah, I want it specifically because of the bugs." So they didn't make me pay for it, which was very nice. Um, and I got to grab a bunch of these little guys off of that, which was quite fun. But then I've also found them on a dead horse that I found on the side of the road in Mexico. So, both extremes, the preserved product side and uh the wild encounters of them. If you find this little guy, is it good or bad? In most instances where people are going to be finding them, uh, and by that I mean like on stored products, they'll also go after taxidermy, so they're collections pests. Most of the time when you encounter these, they would be considered, I would say, bad, even though I greatly dislike saying that certain insects are bad. Um, in most cases, when you encounter these, there are easy ways to treat for them, so it's not like a huge deal. But they do typically damage stored products like hams and meats, and apparently dog bones, um, taxidermy, and things like that. But they're very easy to get rid of. They're not like high infesting things. So if you freeze whatever it is, um, I brought that dog bone home and I immediately put it in the freezer. And then I got up the next morning and I had 3 beetles. Classic entomologist move. It certainly is. I'll do the same thing with like other stored product pests, like these guys. Um, I still have the spider beetles sitting on my desk for when we looked at these a couple of weeks ago. Um, These ones are in chicken manure, so not a food product. But this container is oatmeal and dog food. So when I go to the store, and sometimes I will find uh birdseed or rice or pasta or any sort of product like that that'll have insects in it. Um and I'll take it up to the front and basically do what I did with the dog food. And I was like, hey, or with the dog bone, and I was like, hey, uh, there's bugs in this, and they get super apologetic. They're like, oh my god, I'm so sorry. Like, I I don't know how this could happen. And I'm like, it's cool. Can I have it? And that takes them a minute because they were just freaking out that I was going to like call the health inspector or whatever on them or give them a bad Yelp review. And then I'm just like, yeah, no, can I just like have this though? I don't want to have to pay for pasta that I'm not going to eat, but I do want the bugs out of it. Pet stores occasionally. I wasn't there specifically looking for bugs at that point. I was there picking up crickets for feeding my tarantulas and stuff, and I just so happened to look down and see movement in the bone packaging. Um, so not on purpose. I don't scour the shelves, uh, hoping to find one. But if I ever do, and I'm like, oh yeah, that like I can see that this little box of pasta has beetles in it. I just like, hey, just so you know, I found this. Can I have it? And they're like, yeah, yeah, like we don't want it. And I'm like, this is probably something you don't want, so like maybe go check the other boxes just to make sure, but I'll take this one off your hands, like no worries. And it's worked out great for me so far. I got a big bag of birdseed one time. Um there are a bunch of weevils in it, and I was like, hey, the big thing. I'm like, oh my god, yeah, it was like a 20-pound bag of birdseed. Newsflash: the birds will eat the beetles too. It's fine. It's totally fine. So yeah, that's a strategy that you can use. Your chatter. Uh, if you find bugs and stuff that you don't care if they're in there, you might be able to get it for free. Big brain. Those are the important life lessons that you learn on the eventomology streams. Nicole, you seemed quite shocked about the horse. Um, that one. Uh, now that I think about this is kind of a crazy story. Um, but when I first moved to Arizona, a couple of days, and by a couple I mean like two, 2 days after I moved to Arizona, we had a lab trip that we went down to Mexico. We were in central Sonora for a couple days. Um, and we had pulled over high up in the mountains, and we were just stopping at this place for lunch. We were sitting there waiting for the rest of our group. Uh this was a big group trip where there was like 20-25 cars that were going to be coming with us, that was organized by one of the universities. And so we got down there and we're like, this is the meeting point. We're going to be there at X time. But the other groups that were coming were not all entomologists. We were the entomologist faction, and then like the botanist and the mammologist and the herpetologist and all these other people were meeting us there. And so for them, it's a lot more difficult to like collect specimens along the way. A lot of the time they're not allowed to like actually collect things. They just have to like take photos and measurements and stuff in the field. And so rather than all driving down together, we left a couple of hours early so we could stop along the way and still be at the rendezvous point at the right time. Um, and so yeah, we stopped and we were just poking around, and off to the side of the road there was a horse, or what was left of a horse. It was pretty far gone. Like I said, these show up at like really dry dead stuff. So this was not like a gross super active dead dead vertebrate that the insects were on. Um Once a horse. Yes, there indeed was once a horse. Um so yeah, there were a bunch of these things on it, and that was the first time that I had actually seen them, like I said, in the wild. Um all of these insects that we associate, ugh, sleepy. All of these insects that we associate with like human habitation, and we call them stored product pests and all this other stuff, like they existed before we were storing product. Uh these beetles existed before we had ham. The uh spider beetles or whatever that I have over there existed before there was oatmeal and dog food in a jar for them to feed off of. So, um, the things that they were naturally eating still exist, and the only reason that we notice them in such high numbers is, yes, exactly, Tortilla. We essentially built them buffets. We took the thing that they were naturally eating, like seeds and grain and whatnot, and rather than it being out in a field where they have to crawl between plant to plant to get to multiple seeds, we've now just taken it all and shoved it in a bag. And so now there's a super high concentration where imagine just going to live in a place that's made of food. Like they're just chilling in there, and any direction that you look is something that you can take a bite of. And so they really figured it out after we developed products to store. Um, and they just happen to find their ways in to products that we hang on to. And this clarid is in, as I said, dried meat product. All right, so I think that's probably good on clarids. Let's move to biphilids. Cam. All right, I need to put the labels back on these real quick, so give me just 1 second. They attracted to leather. Um, I bet they would do okay on leather if they had to. And by that I mean leather is oftentimes treated when it's going through the process of being made into stuff that they probably wouldn't go after it as their first choice, but they do go after taxidermy and things like that. So it's not super far off from leather. They're monsters I'm familiar with. Yeah, carpet beetles more often. Um, the ham beetle is like a very minor pest, and most of the time it's not going to be that. Carpet beetles or dermestids are definitely the more prolific and the more numerous things that would be eating that sort of stuff. But it could be a ham beetle. All right. Now, we're moving on from ham to fruit, specifically raspberries. So, this next little guy here is a biturid, and it's on a point, so you know it's quite small. This is the raspberry fruit worm. Don't even own any leather. Ta-da! Does my Why does my white balance not stay the same after I change over? I don't love that. This is a very, very small group. In terms of species, there are only three, I think, in the U.S. Let me fact check myself. Oh, I lied. It's even worse. There's only two. So two species and two genera in our area, 24 species worldwide. So not very speciose at all. Like a bark beetle. I can see it. I can totally see where you're coming from. But let's a slightly different view and start talking about some of his little defining characters. So as I bring it, like I said, this is a claroid, so it is going to have these pretty densely hairy elytra. You can see all of those hairs going down. These are what we would call decumbent, because unlike on the clarids where the hair was sticking straight up and like very poofy and fuzzy, all of these hairs are pretty streamlined, pointing backwards. So our word for that is decumbent. Additionally, if you look at the pronotum on this, so this neck shield area that we're kind of focused in on right now, with the last family, it was narrowed towards the back. With this one, it is very broadly attacked. You can see that that pronotum at the base is almost as wide as the wings are. They are also convex, which everyone have fun going back to geometry. Convex is like bowing out as opposed to concave where a shape bends in. So if we look at this guy from the side, it has a pretty domed appearance. Let me turn this. I had to adjust my here. Jamie, hello. Welcome in. Are we gonna? There it is. So not the flattest beetle by any means. Some of these other things like the biphyllid that we're going to look at next are going to be pretty flat. This guy pretty heavily domed. It's got this very rounded look to the elytra. The head is also deflexed, so if we spin him just a little more, get a the head is looking downward. it's coming out of the beetle, but the mouth parts and everything are directed down. And from this view, you can also see this is as far of a zoom as we have. So that should also give you some idea of scale of this guy. Uh Clubbed antennae again. So right here, right there is the antennal club. And then the other big thing that I use to look to identify this group is another tarsal character that Tortilla's gonna hate. I don't even know if we'll be able to see it. Does he have a lot of eyes or is that just texture? Well, it's one eye. Let's see. Let's go back. So again, this is as far of a zoom as we can get on this. It's a pretty tiny beetle, but I'm assuming there we go. That's pretty in focus. So you're seeing all those little cells on the eye, Jamie. Insect vision works a lot like pixels on a screen. So if you're a blind or if you're like a cave insect where you don't really need to see, sometimes you have like maybe 2 or 3 little eye segments, which are called facets, FACETS, and you might have just a handful of those. Because if you're in a cave, you don't need to have a lot of high contrast vision because there's not much to see in a cave. It's dark, and you're doing most of your foraging and stuff with other senses. But as you get further and further up into insects that are doing a lot more different things and are a lot more active and oftentimes are hunting, you need better and better and better vision. So these guys are not predatory. They're feeding on and laying their eggs in fruit and flower pieces. So this one in particular, this is Biturus unicolor. Um, this is the raspberry fruit worm beetle. And so they are laying their eggs and they're feeding on things in like the bramble family. So that's like raspberries and blackberries and things like that. Um, Rubus, that whole genus, and Geum is the other one. So they're going to be feeding down in the pieces of that. Um, in the dropped, like in the bases of the flowers, that the achenes, the seeds on the outsides of those fruits will oftentimes be fed on by these things. So these aren't predatory. They don't need a lot of vision, but they're still out and active and flying around. So if you only had like 4 pixels, you don't get a very clear picture. That might be enough to see like light versus dark, and you'll be able to differentiate that way. This guy has a lot more pixels where it would be able to make out like plant versus water versus sky versus predator or versus other bug. Um, and so, yeah, insect vision is a lot like pixels on a screen. Something like a praying mantis, though, which give me praying mantis. So excuse me using dreamstime.com and not having something specifically prepared, but Mantids are predators, right? They need to be able to see an insect buzzing by. So they need to be able to differentiate between sky and plant and tree and bird and insect, like much smaller than itself, flying by. And so they have a ton of little segments in their eye because it gives them more and more and more refined vision. Little beetle in your apartment. Did you get a picture? Try to get a picture. Misu Kisu. Hello. Um, but yeah, so the Hollywood thing of like repeating the same image a bunch when they show insect vision doesn't really make a ton of sense. Because like why would they need to see the same thing a bunch? It helps them with clarity to be able to see Prey 180p Predator 4K. Couldn't have said it better myself, Jamie. Yes, that's exactly how that works. We'll send in bug core. Yes, please do. Um, this is not related to the beetle ID that we were going over, but do you know why mantids? Mitsu Kisu, you're reading my mind. Exactly where I was going. So yes, on a mantis, this black dot is called a pseudo pupil. Pseudo is false, pupil is pupil. So it acts like a pupil and it will follow. So depending whatever angle you're looking at the praying mantis at, there will be a dot in the eye somewhere. But it's not always in the same place, but it doesn't move the way you think it does. It's not actively tracking. The really crazy thing, and some people are not going to like this revelation, is that you're looking down into the praying mantis's head. It's like if you're looking down like a tube, and so you can't see the pupil here, but right here, when you're looking straight down the barrel of it, you can see straight through. It's like a yes, exactly, Trilla. It's like a big bunch of straws. So the reason why it looks like the pupil is following you is because that pupil exists where your line of vision is looking into the eye. It's not head empty. It's dark in there, Courtney. What do you want? It's not going to be super well lit. Um, but yeah, so that's the reason why the mantis egg or the mantis eye looks like it follows you, is because the location of where that pupil is completely dependent on where you are. You'll notice that all of these mantises that were looking at the pupil is coming from the camera. Roach, hello. Welcome in. But yeah, so we're looking where the camera is able to see this. And some of them are a little bit more off, but even something like this, where the angle here, the camera is still probably up. What's the function of that tube? It's called the rhabdom, Rhabdum. And it's essentially like our rod and our cone. It's letting light into the back of the head, uh, and then it bounces around in there, and they're able to see things. But since you're looking straight down the barrel of it, you're able to see back into where it attaches. No thoughts. Yeah, head empty. The other cool thing though about praying mantises, this is a real picture from a real scientific article. they can see in 3D. They put little 3D glasses on the praying mantis, so they're actually able to see in 3D because they have such advanced and so many, again, quote, pixels in their eye that they're able to basically make 3D shapes, which is very, very cool. Let's see if it'll let me open this. It does. Exception to very tiny robots. Very cool. But it also Spy Kids 3D enjoy it to the fullest. Yes, they certainly could. Um yeah, so they're able to do this, and if you think about it, it makes sense. Yes, paper jewels, exactly. If you think about their needs and how they hunt, the praying mantis will have its little curled up arms and go out and grab things midair. In order to be able to do that, you need to have some level of depth perception to know how far away from you that prey item is, so that you don't just like, here's the prey item and I'm a mantis, I'm like reaching out and I just can't grab it. You need to be able to make sure that it's within your grasp, that you can go out, get the thing, and bring it back. So it makes a lot of sense that they can see in 3D, but how do you test whether or not they can see in 3D? So my understanding is that these colors actually block some of their let's see if it has. Hide certain dots, causing them to move. Dots would appear to move in one direction. The other eye. Yeah. So they put the dot within striking distance and they attempted to grab it um, with the filters. So they were able to filter out the one color from one eye and the one color from the other eye, and they're able to kind of combine it to be able to grab 3D things. So very cool, very slay. Ring Mantis 3D sets. This this is my favorite picture. This is the one I was looking for. He just looks like he's so chill. Like absolute chillest 3D glasses. Would the sound waves from the cinema kill the bug? Real question. No. So most of the you're about to see me. Well, maybe not see me, but I'm gonna do a Google real quick. Most insect ears are not in their head. Praying mantis ear. Great question. Oh, what? It's on the bottom? Whoa. I did not expect that. So, oh, tortilla, yes, please. New wiki emote, that would actually be very funny. If you need me to send me that picture later, I can. I can find the exact one. Yeah, so apparently the mantids do have an ear on their belly. What I was saying, Nicole, is that things like grasshoppers, their ear is either on the side of the abdomen or on the side of their front leg. And so the here we go. So the sound waves would not like explode the head of the mantis because that's not don't want this. No, I don't want this. Go away. um, this is the ear. So there, Jamie, behave. Um, yeah, this makes sense for mantises, I think. Um, just the way that they're built, their front legs are up off of the ground. So if like a grasshopper had a cricket on the underside of its belly, that's going to be in the ground all the time. But because mantids are basically on four legs, they kind of centaur most of the time. Their front two legs are up. This part between the second and third pair of legs is always going to be exposed because they're so high uh, up off the ground. So that makes sense where a mantis's ear is. But that would not let their brain explode from the movie theme. We can do Erosi if y'all want to do Erosi. I don't. Emotes don't matter. What's it matter? Yeah, I think they probably would only have the one ear. It's it's located pretty medially, so either side probably contributes half to the structure on the inside that allows them to hear. But on the outside, because it's in the middle and like the middles overlap, it's basically just going to be the one structure. Thanks, Liz. Cheers. Um, yeah, by turds. Or no, yes. Yes, by turds. By turdy. Um, what I was saying before we got onto mantises, which totally fine, by the way. Um, Wednesdays we do generally have a plan, but I am always happy to derail that plan if there are questions. So even if it's not related to the current topic or the current specimen, yeah, ask away. But the last thing that we were going to look at was the other defining character that I would use for biturids. And we do have one that is actually pretty visible, which I am surprised about. So this is a very strange tarsal structure. We are looking at the front right leg. So it's basically right here below this corner of my camera. That's the leg that we're looking at. Um, and the thing that we want to see. How do I turn it a little bit more this way? There we go. We've got some white space now. So this is as far in as I can get. But right here, you can see that there's like there's a little piece that's coming off under one of those tarsal segments. That is a biturid character. Now, with the that's an awesome wiki note. I love that so much, actually. Um, there are other insect groups that will have lobed tarsi like this. But this is a unique instance where the first tarsimer is normal. 2 and 3 both have this little lobe underneath, and then 4 and 5 go back to being normal. So typically, what you see when you have a lobed tarsimer or you have this like projection under one of those structures, it's usually going to be the second to last structure that actually has that lobe. This one is weird in that it's only the middle pieces. So again, 1, 4, and 5, normal. 2 and 3 have this projection under them. So if I were looking at this beetle, first thing I would notice, it's hairy. Okay, that maybe leads me into the claroid group. Let me check how many tarsi it has. I would flip it over. I would count 5, 5, and 5. But while I was counting that 5, 5, and 5, I would also notice, oh, this one has lobes, but not on all of them. So this is a very common beetle in the northern parts of the US. They're super widespread, basically all across the north. Um, they're in Washington, they're in British Columbia up there. They do get down into Georgia. I don't know if they're actually known from Florida, though. Might be a little bit too far south. But really neat little beetle. I've only collected them in the wild once, and it was in Pennsylvania on Blackberry. So neat little group. There's only 2 in the US, so uh, I'm halfway to a complete set. The other one, Zeracea, is West Coast. So the family is basically all across the US. But the one that we're looking at here is definitely the most common of them. Hmm. What do we want to? I think we can get. Yeah. Well, I'm making the executive decision. We're going through one more. We're doing one more family. So that was Biturity. Now we're going into Bifilidae. Another little guy. You don't need it quite at 50x yet. There we go. Look at our little guy. Granola's real excited about it. So this is a biphyllid, which are known as false skin beetles. I don't truthfully know where that comes from or why they would be called that. But if we zoom in, this is all the way in, so we're at 50x. This is the most we can do. And you can't even see the. How are you going to see the submental pits? So you can see the hairs on this, right? Again, it's a claroid. They're generally going to be pretty hairy. This one, as opposed to having really fine hair that lays back against the body, I would venture into calling these bristles. They stick out and they all sort of curve back, and you can see it very clearly right now along the edge. But if we go from the side, you'll also be able to see that that hair is like going back. Leaving work, so you're gonna miss part of chat chats. No, that's okay. We have the recaps in the Discord, and there are VODs, so you can always go back to the VOD if you need to, Lyndon. You can also at any point message me beetle insect questions. Uh that is a perk for sure. So from this angle, you can also see those same hairs. They're not flat against the body like the other ones were. These are up. They're spikier. They're like if you just slicked your hair back. My chair just I almost died. That was crazy. Yeah, some peach fuzz. Yeah. Um. So to separate this from the last group, oh, excellent, Vic. Yeah, we can't. I gotta get something that can zoom more. I'm not gonna do that because it's gonna like create totally break the stream setup. But this hind tarsus that's sticking down right now, it's very thin and narrow. So you're not going to confuse this with the last one that had those weird lobes. There are no lobes or anything here. These have just very straight, very thin cylindrical tarsi. So aside from the difference in the hairs, um, you would see the difference there in the toes as well. If we look at the pronotum, and I don't know how well you're going to be able to see them. You heard it? Yeah, I bet you did. I saw the mic spike. You get this full screen so I can as clear as possible, but I don't know if we're going to be able to. I mean, not. Um, this is gonna maybe actually I can find a better picture. Apparently they don't. Apparently nobody's got to have a good picture. That's frustrating. Um, it's it's hard to see, but it is there. I promise. So on either side of the pronotum, there are these two raised ridges. So the design on the pronotum sort of goes like this. There are two carina, Carina, that are raised that go down the sides. I'm going to do my best to point at them, but I don't know that we're going to be able to see them on this. So on the close side, they are here and here. Basically, right where my pinhead is right now is between both of them. You can sort of see the bases. But then on the other side, you might be able to see the shadow of them a bit more. Like right here, give or take. So we haven't run into many of these where it's something that I can see pretty clearly here, but for whatever reason, the stream doesn't pick it up super well. Let me hope maybe the boosted. I don't know that changed it. But source dude, just trust me, there are two grooves or two raised ridges that go down each side of the pronotum. And then if we can't see that, we're certainly not going to be able to see my favorite structure on a biphyllid. Which is the cetos premental pits. Maybe we can. Maybe we can see the cetos premental pits. Okay. Bring it out some more. And I am going. All right. Who's here? Who's with me at the moment? I need some sort of validation here. So right here at the tip of my pin, there's a little hole. You guys see that oval? That's kind of like under the chin. Granola says sure. Nicole says wah. Gabriel says kind. Okay. Slightly greenish. I don't know about green. One second. All right. We're gonna landmark it first. So do you all see here the antennal club? All right. We're at antenna. If you follow the antenna back to the base, you have the eye right here. Yes, we're there. Okay. Directly below the eye. So there's the eye, there's the antenna, and then there's this hole that we're looking at. I'm going to do my best to point at it again. Right. Oh, leave the pin. Oh no. That was a disaster. Okay, we're back. So right here. Yes. Yes. So that in the key, uh, with distinct lateral pore opening into a pre-gular pocket. Do I know what that hole does on, like, basically the chin of this beetle? No, I have no idea. It it could be a mycangium, which we've talked about in some other groups before, but it's an area where you store fungal spores to essentially inoculate where you're living with fungus that you are familiar with. So you basically take a little bit with you, and then you can start that fungus at a new tree. These have been found in fungus. They are like vaguely associated with fungus, but we don't know. Explain whole. I don't know. It could be a place to store fungal spores, and that would make sense because the opening of this hole is surrounded by hair. So it's like one of those things, like a trash can where you can put it in, but it keeps everything closed. Um, it's where you keep your mushroom spores too. That's funny. Blue stain fungus. Yes, yes, exactly. So that is the working guess off of what this structure is for, but I don't think we have confirmed, and I don't think we know that 100% yet. But most other mycangia that you see on small beetles like this, one, the beetle's not going to be super hairy, and two, it's not going to have that like dense ring of hairs around the inside. And so there's not very many biphilids. I think there's only like 5 in the US. But they're really easy to ID because they have those little fuzzy pits. And I think that's neat. Um, they all have that. Uh, I've not seen every biphilid in the world, but I think that is one of those characters that is consistent across the entire family. Now, I know we haven't talked a lot about those tonight, and there's been a lot of this or that or kind of sort of, but I think all biphilids have that hairy pit under the mentum. And so pretty quick, pretty easy ID if you check for that. And this was actually the first biphilid that I ever collected, and it's from this past July. This was in a fuzzy pits are in hell yeah. This was in a flight intercept trap when I was down at the beetle course in Arizona. Um, and this was actually a new family for that class. So for those of you that maybe don't know or haven't heard in a while, um, there is a Beetle ID class where where people will come and learn what you guys are learning on Twitch right now, but in person with other instructors besides myself. But we make a list every year of how many beetle families that our course can collect in the 10-day time that we're there. And so I took the class as a student in 2019, and then I'm still on the instructor team, but I taught it in 2023 and I taught it this past summer. Um and this year, so 2026 now, but last year then, 2025 was the first of the three courses where we actually found biphylids. And it was just that one. The one that you guys were just looking at was the only specimen that we've gotten in the 3 years of having people down there looking for stuff. Um, and I pulled it out and I was like, holy shit. I think we just got one that we've never gotten before. There's a lot of overlap between the classes in terms of like, oh yeah, we always find a ground beetle, we always find a scarab beetle because those things are a lot more common. But the goal with the class is to take the time and look at all of these little tiny things and notice these really tiny differences that we've been talking about doing all of these pre-chat chat segments. Um, and finding all of this diversity because that thing could have been in a box with any other number of beetles until someone looked at its pits. So that's a cool specimen because like I said, that is the first one that I've ever collected, and it was the first one that our class had ever collected over the 3 years that we had done it. So thought that was pretty cool. All right, we are going to take a quick break. I'm going to send y'all home back to Alveus for chat chats in about 25 minutes. If you're not sure what that is, check Alveus out in 25 minutes from right now. And I will be looking around an animal sanctuary in Austin, Texas for whatever bugs may come onto their big fancy cameras. But I'm going to take a little bit of a break. And I will be back, and you will hear me in 25 minutes. I will make one plug now. I'm going to plug it again after chat chats. But one of the bug cord community members is working on a project about insect fears and how you've gotten over them. There is a Google form in the general channel. So if you would like to go answer that and help them out with their project, go check that out for me. You can definitely do it in the interim time between now and regular chat chat. But I will send you back to Alveus, and I will catch up with you guys in about 25 minutes. Bye. Have a nice night. See you soon.